So let's talk about genetics. When Darwin was working on his theory of evolution, the field of genetics did not exist yet. Darwin talked to farmers and people who were doing animal breeding as a way to learn about heredity. People have always probably noticed that children tend to resemble their parents, and at least since the beginning of agriculture 10,000 years ago, people realized that if you selected seeds from plants that had traits that you wanted, when you planted those seeds, the plants that grew would be similar to their parents. And the same with animal breeding. We have been domesticating animals deliberately for thousands of years, and people realized that if you selected the animals for breeding that had the traits that you desired, you could amplify those traits generation after generation. Darwin also learned from talking to farmers that new traits did appear. But heredity was thought of as being some kind of a blending, that because that's kind of what it looks like. When you look at children and their parents, it kind of looks like the kids are a blend of traits from the two parents. And the same was found by people who were doing animal and plant breeding. And so it was thought of as being some kind of a blend. It wasn't known that traits were on the chromosomes or that DNA hadn't been discovered yet. Some traits were known to skip a generation too, particularly things like blue eyes and red hair were known to skip generations. And other traits were known to not skip a generation, that they were present in parent and offspring in every generation. But nobody knew the mechanism of heredity until the 20th century. DNA was not even found to be the molecule that was the mechanism for heredity until around 1950. So before that, the whole field of heredity had they had breeding experiments, but no mechanism, no molecular mechanism was known. So the first person that we consider an actual geneticist, or the first geneticist, is Gregor Mendel. And Gregor Mendel was an Austrian monk. He did most of his work in the 1860s. So that's around the same time that Darwin was publishing his first works on evolution. Mendel, though, was not a scientist. He was a monk who wanted to be a teacher, but he failed the teaching exams, so he was not a teacher. He eventually became the abbot of the monastery where he worked, and he was really interested in heredity and specifically hybridization, the crossing of different strains of plants. And the field of genetics didn't even exist when he was doing his work. So he was working in isolation. He wasn't a trained scientist. He wasn't in contact with other scientists, and his work was almost completely lost to history. It was just by chance that somebody in the 1920s who was interested in genetics came across his papers in a German agricultural journal, and it was rediscovered, and their importance was realized. So Mendel did plant breeding experiments using garden peas, and peas have a few advantages. One was that he could order lots of varieties right out of a seed catalog. Backyard gardening was very popular in the 1800s, and there were seed catalogs, and so he could just mail order different varieties. And since he was interested in crossing different varieties and hybridization, that would have been an advantage for him. Peas also have a really short time to set seed. If you buy any kind of a plant, they always have a number of days to maturity, and for tomatoes, it's like 80 or 90 days to maturity, and peas are much shorter than that. So in his high altitude monastery, which had short summers and cool climate, he was able to get 3 generations a year of peas, and that is really impressive. As a gardener myself, getting 3 generations of a plant in 1 year is very impressive. So he knew that in peas, that from he did some preliminary experiments as well. We don't have his notes, unfortunately. All we have are his published papers, because the abbot who took over the monastery after him was worried that they were going to somehow be taxed for his farming experiments, and so he burned all of Mendel's lab notebooks and all of his correspondence and all of his letters and all of Mendel's papers were burned after his death. So the only thing we have are two published papers to know what he did. He knew that traits disappeared and reappeared. He must have done preliminary hybridization experiments for his peas, but we don't know exactly what he did. And also in peas, you can control fertilization. So in plants, plants have male and female gametes, like animals do. The female gametes are found down here in an ovary down here, and they're called ovules, and they are fertilized by pollen. And the pollen, which is produced in the anther here, so most plants that you're familiar with produce both pollen and ovules. And they would prefer not to self-fertilize most of the time. Some plants have mechanisms to prevent self-fertilization, but they can self-fertilize often. And that's the case with peas. So with peas, their own pollen can fertilize the ovules. And there's a petal that completely covers the sexually reproducing parts of the plant, which is important because that prevents cross-pollination. That keeps out bees and other insects that might cross-pollinate. So normally, plants produce showy flowers to attract pollinators. If a plant does not have a pollinator, like wind-pollinated things like grasses, you wouldn't even know they had a flower because it's not meant to attract a pollinator. But flowering plants that have a showy flower, it's meant to attract a pollinator. And peas are kind of odd in that they have a showy flower, but they have this petal that covers up the reproductive parts of the plant, so there's actually no pollen available for, or nectar available for pollinators. So, but normally, in a normal showy flowers, this flower would be open, a bee or something would come in, drink nectar, which is usually down here at the base, get dusted with pollen, and then move on to another flower, so that each flower is getting pollinated by, the ovules are getting pollinated by pollen from different flowers. So that's the purpose of the showy flowers. So in peas, you can prevent that from happening by just leaving it alone and it will self-pollinate, or Mendel could cut open this enclosing petal, remove the anthers before any pollen was produced, and then he could fertilize the plant with whatever pollen he wanted. So that was an easy way to do cross-pollination. Now, once he did this, he would have to cover up the flower to prevent bees from bringing pollen in or wind from blowing pollen in. And so he probably used what most botanists have used to control fertilization, and that is little paper bags over them, little wax paper bags to prevent outside pollination. And so the peas in a pea plant, this is already the next generation. So the whatever trait the pea itself has, that's a trait of the offspring. The pod is a trait of the parent plant. So whatever color the pod is, or characteristics of the pod or the plant, that's the parent generation. The peas, the characteristic of the peas is the next generation already. So whether the pea is green or yellow, that's a trait of that next generation. So we still use some of Mendel's terms today. So he called his plants that he initially hybridized, his two different varieties of plants that he hybridized, he called those the P generation for parental, and we still use that today in genetics. And then he called his offspring, he had a first generation of offspring and a second generation of offspring, which he called the F1 and F2 generations, for F for filial, which is the from the Latin word for sun. So when we still use that today in genetics, the P generation, the F1 and F2 generation. The F2 generation, that's like the grandbabies of the original parental generation. So this is a picture from a Victorian seed catalog. So this would be the different varieties that he would have to choose from. So what he found with his initial, which we can assume that he did some initial crossbreeding, sorry, that's my dog, some initial crossbreeding experiments, we can assume that he must have done some of those. He picked varieties that he called true breeding varieties. So true breeding variety, that would mean that if you let it self-pollinate, that you will get nothing else but that particular variety. So for example, if we think of dogs like Chihuahuas, if you have 2 Chihuahuas and you let them mate, you would expect all the puppies to also be Chihuahuas. So if it's a purebred breed like a Chihuahua, you would expect it to be true breeding, meaning that when you cross them, you don't get anything else. And that's not Darwin. Mendel meant the same thing by his true breeding varieties, that if you cross them together, you would just get the same trait. You wouldn't get new weird traits appearing that weren't present in the parental offspring. So that's what he started with. He ordered seeds from catalogs. He did some experimental test crosses, and then he picked traits that were true breeding varieties. So his experiments that he published, so he was interested in hybridization. What happens when you cross 2 different varieties? So one of the traits that he used was flower color. He found a purple flowering variety and a white flowering variety that were both true breeding, meaning if you cross them to each other, you would just get nothing but purple flowers or nothing but white flowers in the next generation. So his true breeding parental varieties we call the P generation, and the next generation, the hybrid offspring of those parents, we call F1, and we get that from Mendel. And then when you let the F1 individuals self-pollinate or without crossing them with anything else, we call those the F2 generation. And we still use those terms in genetics today. So the other thing that he was looking for was he was very interested in traits that disappear and reappear. So it was known that in various kinds of animals and plant breeding that there were traits that would disappear and reappear. And so that's what Mendel was interested in. And the reason that we consider him the first geneticist is because not only did he notice this, that traits disappear and reappear, but he counted the number of individuals that you got, and that led him to some conclusions which were very insightful and, more importantly, correct. So what he noticed, what the traits that he looked for, he wanted one of the traits to disappear when he hybridized them. So for his purple and white flowers, when he crossed them, he got all purple flowers in the F1 generation. When he planted those seeds, all the plants had purple flowers. So the white flower trait disappeared in the F2 generation. But when he let these purple flowers just self-pollinate, in the F2 generation, then the white flowers reappeared, and in a very specific ratio, in a 3-to-1 ratio. So in the F2 generation, he got 3 purple flowers for every 1 white flowered plant. So one plant only produces one kind of flower. The plant is either a purple flowering plant or a white flowering plant. So, and this is what he was looking for, and he managed to find 7 traits in peas that behave this way, where when you hybridize them, one trait disappears, and then you can get it to reappear in a 3-to-1 ratio in the F2 generation. So this is what those purple and white flowering plants actually look like. So Mendel called the purple flower and any of the traits that never disappeared and reappeared, he called those dominant. And we still use that today too, as well as calling the one that does disappear. We call those recessive. And here's another case of an old-fashioned word that has gotten frozen in science. Recess, something that is recessed or recessed is something that is set back. And that's the sense that he was using this word. So to him, recessive meant that it didn't disappear, but it was set behind. It was set back from the dominant trait, which never disappeared. It was covered up by the dominant trait. But his important insight was that it doesn't disappear. It's not being diluted. There isn't a blending going on here, which is how most people viewed genetics at the time. Instead, that recessive trait was there, undiluted, and he could get it to reappear in the F2 generation. All right, so Mendel had several really good insights, which is why, even though he only had 2 published papers, we consider him to be so important. So he realized that these traits were not actually disappearing or skipping a generation or blending. He realized they were there all along, just being covered up. The recessive traits were just being covered up by the dominant trait. And this led him to the realization that there are discrete units of heredity controlling each trait. And individuals have 2 copies of these traits, one from each parent. And they're not blending, and one might be covering up another one, though. And that's what he called the recessive traits. So those were 2 really important insights. One of the analogies that he used was that these are like letters in an envelope. You can, the hereditary factors, what we call genes and alleles today, he called heritable factors, that these factors are like letters in an envelope, and you can put one envelope on top of another envelope and cover up the letter, but it's still there. It didn't disappear, and that if a trait seems to be skipping a generation, it might still be there, just being covered up by the dominant trait. So the word gene didn't exist yet, genetics didn't exist yet. Those words would not be coined until about 50 years after Mendel did his work. So the 7 traits, this table shows the 7 traits that he found that behaved this way. So here's the purple versus white flowers that he used. So on the left, we have all of the dominant versions of the traits, and on the right is the recessive form. So these were separate traits. These were not all in the same pea plants. These were different varieties of peas that he ordered. And so he found, besides the purple versus white, yellow versus green. So the one on the right, the trait on the right, when you cross it, that trait is the one that disappears in the F1 generation. So yellow seeds is dominant over green seeds. Round seeds is dominant over wrinkled seeds. And you notice for the seeds, here are his actual counts from his published papers. There are way more. He has thousands for the seed traits, but only hundreds of the others. And that's because for these other traits, flower color, pod shape and color, he had to actually plant the seeds and then see what trait was in there in the grown plant. But for the peas, the characteristics of the peas themselves, that's already the trait of the offspring. So yellow versus green, he didn't have to plant them to see whether they were yellow versus green, because you could just look at them without planting them. So that's why he has so many more of those. And then for all of these, he got an approximately, in the F2 generation, a 3-to-1 ratio of the dominant to the recessive. So round seeds versus wrinkled seeds, green pods versus yellow pods, inflated pods versus constricted pods. So a constricted pod, that's like snow peas, where you can see the peas inside the pod. Axial versus terminal flowers. So axial flowers means the flowers form on the main stem of the plant, and terminal flowers is the flowers are on the ends of the branches. And remember, it's the flowers that eventually turn into the pea pods. So the reason why a plant with axial flowers might be in a seed catalog is because all those heavy pods that would form later wouldn't be on the ends of branches, they would be on the main stem, which would mean that this plant wouldn't need to be trellised. Peas normally need to be trellised. They have to grow up against a trellis or they fall over onto the ground. So this would be a variety that would be desirable because you wouldn't have to trellis it. The same with dwarf plants. So tall plants versus dwarf plants. The dwarf plants as well wouldn't need to be trellised. So that's why those would be in a seed catalog as a trait. So now there were other P traits that didn't behave this way, that showed things that look like blending, and that he didn't pick those. He specifically picked traits that fit the quality that he was looking for, which is he wanted a version of that trait that would disappear and reappear, so he could count them. So he could count the number of disappeared and reappeared ones. So here's a summary of what his experiments look like. So for all of those 7 varieties, 7 different traits, he did the same experiment. So here's our parental true breeding types. He crossed them, and it didn't matter. He showed that it didn't matter if the pollen came from the dominant one or the pollen came from the recessive one. The results were the same. So F1 generation, they all show the dominant trait. F2 generation, we have dominant versus recessive in a 3-to-1 ratio. And the dominant ones aren't all the same. So when he let these dominant ones here self-fertilize, 2/3 of them were like the F1 generation. So the recessive ones were always true breeding. There was never another trait hiding in there. And only 1/3 of the F2 generation dominants were also true breeding. The other 2/3 were just like F1. And if you let them self-pollinate, you would get, again, a 3-to-1 ratio in the offspring. So these were the results that led him to his important insight that each individual here has 2 copies of whatever the trait is. And when gametes are formed, only one of those copies goes to each offspring. And so here, when you see the F1 generation is dominant because one parent had the recessive trait, that means that in his F1 generation, we have 1 dominant trait and 1 recessive heritable, or I should call them heritable factors. 1 dominant heritable factor and 1 recessive one in all the F1 individuals. So when they reproduce, each gamete has a 50% chance of having either the dominant one or the recessive one. And how that ends up as a 3-to-1 ratio in the next generation, we will look at that. Okay, so there are 2 ways to breed these pea plants. One is to just let them self-pollinate. So if it's a true breeding variety, you won't get anything but 1 trait. So if it's a green pea, that's the recessive one, remember? So recessive ones are always true breeding. You get nothing but green peas in the next generation. If you have a yellow pea and you plant it and it was a true breeding dominant, then the next generation you get nothing but dominant yellow peas. But if it was from the F1 generation of one of his experiments or one of those that's not true breeding, then you're going to get, the next generation, you're going to get approximately a 3-to-1 ratio. Of course, not in every pod like this. You would have to look at hundreds and hundreds of peas to get that 3-to-1 ratio. They're just showing it here in the 1 pod, just to give you the idea. The other thing you can do with peas is hybridize or cross-fertilize them. So you take the pollen from one plant and use it to fertilize the flower of another plant. And if you do this with the 2 parental types, so the recessive one is always true breeding. If the dominant one is also a true breeding, this is like the F1 generation, you're going to just see the dominant trait. If, however, you take your recessive true breeding variety and you cross it with the F1 generation plant or the F2 generation that's not true breeding, now you're going to get 50% yellow, 50% green. And we're going to look at how we figure that out today. So Mendel's important contributions. One is that parents transmit traits by discrete heritable factors. Now, he did not know that these were DNA, and he didn't use the word gene. He called them heritable factors, and he used the German word for factor. Ah, that's the dog, which sounds very much like the word factor in German. We now call those genes, and different versions of a gene are alleles. His other insight was that each individual has 2 copies of those heritable traits. 1 came from each parent. And if we now use the words homozygous and heterozygous to describe these, the 2 copies that any individual has. So if an individual has two of the same. Homo here means the same. So if they're homozygous for a particular trait, that would mean that they have the same, two copies that are the same of a particular gene. If they're heterozygous, hetero means other. So heterozygous means they have two different versions of a specific gene. So different forms of a gene are alleles. And allele is just from the Latin word that means other or different. So they are, genes have many alleles, often many more than just two. So if an individual has two alleles that are the same, like if you have two alleles for blue eyes, then you are homozygous. If you have one allele for blue eyes and one allele for brown eyes, then you are heterozygous. Mendel realized that these genes are being passed unchanged from generation to generation. It's just that their expression, what we see, depends on which alleles are present. So, and we call how these genes are expressed the phenotype. So phenotype is what we see, how the genes are expressed. And a phenotype could be something that's really obvious, like hair color, skin color, eye color, your height. Those are all parts of your phenotype. Or it could be something else that we can measure, like how fast can you run 100 meters? What's your blood pressure? What's your resting heart rate? What's your fasting glucose? All those things are also part of your phenotype. Genotype is which alleles you have. What is your DNA? What alleles do you have? That's your genotype. So there are other things that contribute to phenotype other than alleles. Environment is important for the expression of certain genes, but that's still part of your phenotype. So phenotype is how everything is being expressed. What do we see? What is visible or measurable as part of an organism's physical form? And then the genotype is which alleles are present, which DNA is present. So if we go back to what the last chapter, meiosis, now we know what Mendel didn't know this, but we know now what Mendel was talking about with each trait has two copies. One came from each parent. We now understand that as being because of the homologous pairs of chromosomes that each individual has. And an individual might have two alleles that are the same. So remember, the homologous chromosomes are not identical. They came one from each parent of that individual. So 23 of your chromosomes came from your mother, the other 23 came from your father. And you may have the same allele on a particular chromosome, and in that case you would be homozygous for that gene. If you have two different alleles, then you would be heterozygous for that specific gene. The sister chromatids, now this diagram here is showing chromosomes that have not yet replicated. So each chromosome only has one chromatid, but chromosomes could have two chromatids if it's after S phase. So once these chromatids, the chromosome has replicated its DNA, then we would have the same allele. So the two identical sister chromatids are always going to have the same allele because they're made by DNA replication. So the genes that Mendel studied are well understood now on a molecular level, and here's just one of them. So his purple flower allele, the gene itself codes for an enzyme, that one of the enzymes that makes the purple colored pigment. And the enzyme is a protein. The white allele is simply a mutated form of that gene. So the white allele, there's a mutation in the gene and the enzyme is not made, and there's an absence of purple pigment for that reason because of this missing enzyme. So this is what we would call a null allele. So let me write that here. So we would call this kind of allele where it's mutated, we would call that a null allele. It's hard to write with a mouse. A null allele is one. Null means zero. which you might know. So a null allele means that nothing that works is being made. So either nothing is being made at all, or the gene product that's being made is useless. That's a null allele. And many, many, many recessive alleles are null alleles because they are mutated versions of the normal allele. And that's the case for our white flower allele. So if an individual has 2 copies of the purple flower allele, they're going to make purple pigment enzyme and the flowers are going to be purple. If the pea plant is heterozygous and has 1 purple allele and 1 white allele, that they're going to have half the amount of enzyme. That's still enough to make plenty of purple pigment. So we can't tell them apart. The heterozygotes and the homozygotes look the same for the purple flower, which is why the parental generation and the F1 generation look the same. The parental generation are the true breeding purple parental type were homozygous for the purple allele. The F1 generation were heterozygous and had 1 purple allele and 1 white flower allele, which is again a null allele. So here's another way to think of this. So genotype we can describe in 2 different ways. We could either give the alleles directly, and normally we use the capital letter for the dominant allele, or if it's a group of letters, we might even use capitals. And we use a lowercase for recessive. So we could either say this individual is capital Y, or big Y, or we could say they're homozygous dominant. Both of those describe the genotype. They don't tell us anything about what this individual looks like. The phenotype then is what we see. There's yellow peas. Heterozygous as well, we could say big Y, little y, or we could say heterozygous. Now for the phenotype, again, that's what we see. So in this case, you can see the phenotype is exactly the same here, but the genotype is different. And notice that the only way we can get the recessive phenotype is if that individual is homozygous. So if we have an individual expressing the recessive phenotype, we know their genotype because there's only one possibility. So if you have blue eyes, which is the recessive phenotype, then we know your genotype. You are homozygous for the blue eye allele. Mendel figured out that these 2 copies of the heritable factors were separating during gamete formation, and each gamete had a 50% chance of having each of those 2 alleles that were separated. And we call this Mendels Law of segregation. And I'm going to put it in air quotes because I think this is another thing that probably this got stuck in early biology, and it's still there. But so a law in science is supposed to be above a theory. So a law is supposed to be like the laws of thermodynamics or Newton's laws of motion, something that's an immutable property of nature that we just stumbled upon. And when it was the early geneticists had rediscovered Mendel's work and were first doing these kind of genetics experiments, they thought they had stumbled upon such a law like Newton's laws of motion. But really, it should be called Mendel's general rule of segregation, because really now we know there are many exceptions to this. And like almost everything in biology, you can have this general rule, and then there's these things over here that don't follow that general rule, and this is no exception to that either. So we call it a law, but keep in mind this is not a law in the same sense that the laws of thermodynamics are laws. I think this was a case of early biologists getting a little carried away and wanting biology to be like physics. And it's not. Biology is way more squishy than physics, and a lot more exceptions to our general rules. So anyway, okay, so Mendel's law of segregation is simply this, that the alleles separate during gamete formation, or segregate. Segregate just means separate. So Mendel's law of the separation of the two alleles, they go into the gametes as just one allele. So 50% of the gametes are going to get one allele, 50% of the gametes are going to get the other allele. So if we have our flower here that's heterozygous for the purple and the white flower color, 50% of the gametes are going to get the purple flower allele and 50% are going to get the white flower allele. So what that means is, so remember we had our phenotype ratio here of in the F2 generation, the white trait reappears and we have a 3-to-1 ratio. Well, what we actually have is 3 different genotypes, not just 2, because remember the purple flowers were different. There were 2/3 of them were just like the F1 generation, and if you let them self-fertilize, you would get again a 3-to-1 ratio. So what we ended up with the genotype is what's called the 1 to 2-to-1 ratio. You get 1/4 of them being homozygous for the dominant trait, 1/4 of them being homozygous for the recessive trait, and half of them being heterozygous and having one purple allele and one white allele. So the way we can figure out what's going to happen in a cross like this is to use a Punnett square. And Punnett is somebody's name. That's why it's doesn't look like any other word. So the Punnett square lets us estimate what percentage each of the offspring, what traits are going to be found in the offspring based on what's going to end up in the gamete. So in the example here, both parents are heterozygous. So this is what would happen in our cross of the F1 generation. So capital P is for the dominant purple, lowercase p here is for the white allele, the recessive white allele. So what we put on the top here and on the sides are the gametes from each individual. So one parent is here, the other parent is here. And we have half of the gametes will have the dominant P allele, the purple allele, and half of the gametes are going to have the recessive allele. And then the way we figure out what's going to be in the offspring is you simply bring down these into the squares and then across from the other parent here. So this square here, we have a dominant here, dominant here. This square, we have one dominant and one recessive. This one here as well, one dominant and one recessive. And this square over here, 2 recessives. So this Punnett square lets us predict that our average percentage here, we're going to get a 1 to 2 to 1 ratio of genotypes, but these 3 squares are all going to have purple flowers because purple is dominant. So 75% purple, 25% white. And this is probability. It's not exact. So the more you have, the larger the numbers, the closer you're going to get to this actual ratio. So for probabilities, we have rules that help us figure out what to do with the information in this Punnett square. So the multiplication rule is for independent events that occur one after the other. So if we have, what are the chances that if we randomly pick 2 P's in a row, that they're going to both have the recessive trait? We would multiply those chances together. If they're all occurring in the same event though, so if we want to know what are the chances that we will have an offspring from the F1 generation that are heterozygous, then we can add these 2 squares together. So this this heterozygous here are the same, so we can add them together. So we can say there's a 1/4 chance or a 25% chance of having a double dominant. There's a 25% chance of double recessive, but a 50% chance, or a 1/2, if we add a quarter and a quarter, we get a half. There's a 1/2 chance of having the heterozygous individuals. But because it's like a coin flip, we have to deal with the rule of large numbers. So in probabilities, the higher your numbers, the closer you're going to get to the expected ratio. So if we just flip a coin 10 times, you might get, even though there's a 50% chance of heads and tails, if you just flip it 10 times, you might get all heads or all tails, or 6 heads and 4 tails. But if we flipped a coin 10,000 times, we would get much closer to that expected 50% ratio. And the same is true in genetics. If you look at just a single small population, like, for example, the chance of a baby being male or female is pretty close to 50%, 50-50. So if we just look at one family, though, you might have a family where there's 4 boys or 4 girls in the family, and another family where there's a family of 4 where they have 2 boys and 2 girls. So you're not going to always get that expected ratio if you have a small number. But if we looked at an entire town of 50,000 people and looked at all of their offspring, we would be much closer to that 50-50 ratio. So the addition rule, this is what I just said. So if you're talking about 2 squares within the Punnett square, it's a single event, and their 2 things are the same, 2 squares are the same, then we can add them together. So the 2 25% heterozygous individuals, we can add those together and get 50% expected for heterozygous. So here's Mendels crosses again with the Punnett square. So here we have our parental types, the homozygous or true breeding white, homozygous or true breeding purple. They only have one type of gamete. We have the purple allele in all the gametes here, the white allele in all the gametes here. So 100% of the offspring are going to be heterozygous in that case. Now, if we take those heterozygous individuals and just let them self-pollinate, or you could cross them to another heterozygous individual, you'd get the exact same result. Now, half of the gametes are going to have the dominant allele, half of the gametes are going to have the recessive allele. So if you let those cross and then you plant the seeds and see what the offspring have, that's where you get this 75% purple to 25% white. But if we look at the genotype, it's 1:2 to 1 ratio. So here's the same thing, but with the pea color. So we have our heterozygous parents here. They're expressing the dominant trait. Yellow pea color is the dominant. And this shows what happens when your graphic designer doesn't know anything about plant biology. We have little swimming sperm here with tails. Representing the male gametes here, when actually pollen for almost all plants do not have flagella like sperm. And these really look a lot like mammal eggs, not ovules in plants. But anyway, it works the same. So we have 50% each for the gametes here. Half have the dominant, half have the recessive, dominant, recessive, and we get that same 3:1 ratio of phenotype, 1:2 to 1 ratio for genotype. So if we have an individual expressing the dominant trait, we don't know whether they're homozygous or heterozygous. And in the past, before we had any kind of DNA analysis available, which is most of the history of genetics, the only way to tell whether your individual was homozygous or heterozygous was to do a test cross. So a test cross, you take your dominant individual here with the purple flower color, or in this case, a horse with a black coat color, and you cross them with a homozygous recessive individual. So for the peas, that would be the white flower variety. And then you see what the offspring are. And if you get all dominant offspring, then that tells you that this individual was probably a true breeding dominant individual. If you get 50% recessive, 50% dominant in the offspring, then that tells you that your individual was heterozygous. And in fact, Mendel also did this. That's how he figured out that his F2 generation, he actually had 2/3 that were just like the F1 generation. So we have our mystery individual here who's expressing the dominant trait. Of course, if they're expressing the recessive trait, we don't ever have to wonder what their genotype is. If they're expressing the recessive trait, then we know they're homozygous for their recessive gene, because that's the only way we would see the recessive phenotype. So here we have our mystery individual. If they turn out to be homozygous dominant, all the offspring will have the dominant phenotype. The only way we are going to get any recessive phenotypes in the offspring is if the dominant parent is heterozygous. So this is a really easy way to tell if we have an individual who is homozygous dominant or somebody who is heterozygous. And we're going to even see this again when we start talking about human traits, because for many human traits that show dominant and recessive qualities like this, we only know what trait somebody's parents had, what their siblings have, what their offspring has. We don't have any way to actually test for those traits. Nor would we want to if they're not medically significant, but we can often figure out if they are expressing the dominant trait, like brown eyes, are they heterozygous or homozygous based on their parents or their offspring. So we'll talk about that later in this chapter. All right, so here's another law from Mendel. The law of independent assortment. And again, this is called a law, not because it really is a law in the sense that physics are, but because the early biologists got a little overexcited about the possibility that they had finally discovered laws that were like Newton's laws of motion. But really, we should call this one as well Mendel's general rule of independent assortment, because it usually occurs, but you know, this is biology. So the independent assortment, this is exactly the same thing as independent assortment of the chromosomes during meiosis. Mendel didn't know why his different traits, when he had multiple of these different traits in his pea plants, the traits sorted independently. We now know that it's because they were on separate chromosomes, but Mendel didn't know that. We now know that it's because when those pairs of homologous chromosomes line up during the first division of metaphase, the paternal and the maternal chromosomes can go to either daughter cell. So the gametes are going to have a mixture, the haploid gametes are going to have a mixture of the paternal and the maternal chromosomes that the parent cell had. So if we look at a dihybrid cross, so the purple and white flower crosses that I've been showing you, that's what we would call a monohybrid cross. There's only one trait. But if we're looking at two traits at the same time, dihybrid cross. So here we have two traits. We have the yellow and round. And here we have green and wrinkled. So yellow and round are both dominant. Green and wrinkled are both recessive. So it's di because there's two traits that we're looking at. So each of these is only going to produce one kind of gamete, one that has, so the Y stands for one gene, the yellow versus green, and the R stands for the other gene, round versus wrinkled. So lowercase r is wrinkled, lowercase y is green. So there's only going to be one type of gamete because these are both homozygous. But the offspring, so now we have our F1 generation, they're going to be heterozygous for both of these traits, but they're going to still show the two dominant traits. So if these traits sort independently, if they show independent assortment, we would expect to get offspring like this, because there would be 4 different types of gametes that could be produced. And so you're going to get this famous phenotypic ratio of 9 double dominant to 3 to 3, each with 1 dominant, like yellow and wrinkled or green and round, to 1. Because we'll have, this is still a Punnett Square, it's just for 16 different squares here instead of just 4 for a dihybrid cross. So if we cross these F1 generation and we get all of these different offspring in this ratio, 9 double dominant, 3 with 1 dominant and 1 recessive, another 3 with the other dominant and 1 recessive, and only 1 out of 16 double recessive, then that means that these are sorting independently. They're on different chromosomes, and so when meiosis occurs, those traits are sorting completely independently. If they are not sorting independently, then we would expect to get something just exactly like a Punnett square for a single trait. And we'll talk about this in a while, that in fact, sometimes we get something that's halfway between these two because nothing in biology is simple. The way that we understand independent assortment goes back to what we talked about in meiosis. So here's our diploid parent cell here that's heterozygous. So we have the dominant R trait here and the recessive trait here. So if this was round and wrinkled, so this individual is heterozygous for that one. On the other chromosome, we have the Y gene, yellow versus green. And I know with the Y especially, it's kind of hard to tell that this is supposed to be lowercase and this one is the uppercase. So heterozygous for both, and there's 2 possibilities during meiosis. Either the 2 dominant traits are going to stay together during that first division, or you'll end up with 1 dominant and 1 recessive when the gametes form. So that's how we end up with 4 different possibilities for the gametes for these traits. Crossing over, if we have 2 of these different genes that are actually on the same chromosome, we do see them sort independently for some of the offspring, and that's because of crossing over. So if crossing over occurs between 2 genes, then we can see a mixture in the offspring. We will see some of these recombinant chromosomes in the offspring, and we'll talk more about that in the next section later in this. Later in genetics, we will talk about how experiments were done mostly with fruit flies to prove that crossing over was actually occurred before crossing over was ever seen under the microscope. It could be detected by looking for this, by the fact that if you did a dihybrid cross for certain traits, you didn't see either of those 2 Punnett square types. In fact, what you saw was something in between those 2 where there was a small number of recombinants evident. But we'll talk about that later.